Hello, everyone. Thank you, Emmanuel and Lauren, for the beautiful introduction. We'll be beginning with the session in 10 minutes from now. Thank you for joining. I would like to welcome all those participants who have joined the session now. We will be beginning with today's webinar in 7 more minutes from now. And we would like to welcome you all. Thank you for joining. And meanwhile, we begin with today's webinar. It would be great if you could please introduce yourself in the chat box, tell us about your educational background and your research interests. Looking forward to it. Thank you. A very warm welcome, everyone. I would like to welcome you all to this webinar on bioinformatics for infectious diseases. This program is dedicated to the study of viral diversity and its role in epidemic infectious diseases that keep re emerging, including zoonotic spillover, transmission between humans, and the process of viral and bacterial disease development. The participants of the program will get a chance to learn bioinformatics and analyze genomic data by applying various analysis approaches to study viral genomes. And as a result, you will learn to understand relationships between viral strains and haplotypes, find differences in sequence data, and see the implications for drug and vaccine design. This program will provide opportunities to practice analyzing data to gain hands on experiences with curated datasets from public domain collections guided by experts with bioinformatics experience and knowledge about virology. To introduce myself, first of all, my name is Sonalika Ray and I'm the Omics Logic Community Manager. I'm responsible for daily interaction with thousands of users that are a part of our bioinformatics community that is managed by Pine Biotech. For those who are joining us for the first time, let me quickly introduce us. We are a US based bioinformatics company who is working with multiple academic and commercial collaborators to develop easy to use analytical tools, and our mission is to make bioinformatics more accessible. Omicslogic research-based training programs are a result of close collaboration between Pine Biotech and numerous academic institutions that have participated in content evaluation, curriculum review, and project design. Omicslogic training has been completed by over 10,000 participants from 162 countries in over 300 workshops. And due to this fast growth, our team is working with local and regional coordinators that are helping refine the local program logistics and leverage our online training resources, adapting them to the needs of students and researchers around the world. The programs follow a project based learning approach using research grade tools to analyze data from top peer reviewed journals. The training has been completed by in six different specialization tracks, which includes oncology, infectious diseases, precision medicine, neuroscience, data science for biomedical data, and comprehensive training on omics data analysis. So, this was about us and our motto behind the training programs. Now, let's start by signing up to the Learn.omicslogic portal. This educational portal will help you get started on your bioinformatics journey. And as a part of this program, the participants will have the access to the Omicslogic courses, which includes courses on bioinformatics, precision medicine, multi-omics, including genomics, transcriptomics, metagenomics, and epigenomics, data science that includes bioinformatics using R and Python, and the structural aspect of biology, that is cheminformatics. As a part of the program, you would also have the access to the example projects of different fields which you could learn from and replicate for your research work. Throughout the course, you will be given directions to access the T-Bioinfo platform for bioinformatics processing and analysis of transcriptomics data. The platform includes demo pipelines as well as data management and analysis cloud infrastructure to run bioinformatics pipelines. So now let's go to the learn.omicslogic portal and sign up quickly. That will help you go through the various resources that our mentors will be taking us through in today's webinar. So I request you all to please put in the link of the portal learn.omicslogic.com on your laptops or if you're connected via your mobile phones. And make sure that you sign up on the portal and have an account for yourself to access the various resources. Now, those who have already created their account on the portal can let me know by putting in one or yes in the chat box, and others can go through the portal and create an account for themselves. We'll be giving them a minute or two to do the same. Let me also follow the steps to assist you. If you click on the login button, you will be redirected to sign up on the portal. Either by creating an account yourself, you can just simply put in your details and create an account, or you can also log in via any of your social media accounts. Thank you, Ashwini, Jamil, and Irandra for quick response in the chat box. Others who have also completed their sign up, please let us know in the chat box so that we can proceed further. Yeah, I see a lot many quick responses. So for those who have completed their sign up on the portal, the next step for them is to complete their profile. So you need to click on your mail address. This will redirect you to your profile tab, where you need to ensure that you have an updated profile picture. Your social media accounts should be connected, and also add a brief bio about yourself. It could include your educational background and your research interests. For every coursework that you complete on the Omixlogic Learn portal, you will be getting the points that you can review here that will take you from a beginner's level to an advanced level. And being a part of the Bioinformatics for Infectious Diseases program, you will be able to see a badge on your profile tab that will ensure that you have the access to the program page and the resources associated. Now, before I hand over the stage to our mentors to discuss more about the curriculum, I would like to launch a poll for all of you so that we can know about you all, your educational background, and your research interests as well. I hope the poll is visible. Ujasvi, can you please confirm?
Yes, ma'am.
Thank you. So my first question here is, what is your educational background? Are you a graduate, a postgraduate, or a PhD or a postdoc? What all are your research interests from the options mentioned here? Is it infectious diseases, oncology, precision medicine, agriculture, neuroscience, or space omics? Also, what is your level of understanding about bioinformatics for infectious diseases? Do you consider yourself to be at a beginner level or an intermediate or advanced? My fourth question is, what type of infectious disease data analysis do you want to learn? Is it NGS transcriptomics or NGS metagenomics or genomics or machine learning for NGS data or structural biology data? And my last question is, what do you seek from this program? Online infectious disease and data analysis training only, or also a research component along with the training, or mentor guidance and one-on-one support? Or would you like to work on an independent research project after the successful conclusion of the program? We'll have the poll on for more 30 seconds. Thank you for the answers. If you have answered the poll, you can make sure that you complete your profile till then. I'll just hand over the stage to our mentors in 30 seconds from now. Okay, so now I would like to invite Mr. Ilya Brodsky to please take over the stage. Over to you, Ilya.
Thank you very much, Narika. Are we going to end the poll?
Yes, I have done that.
Okay. All right, let me share my screen. So thank you everyone for joining today. It's exciting to be here. This is one of the programs that I think is very relevant to what's going on all around us. So it's not unusual that we have so much interest. Let me briefly introduce why bioinformatics for infectious diseases. I mean, why do we need a special program that is called bioinformatics for infectious diseases? How do we think about infectious diseases compared to other types of diseases and conditions? And then I'll briefly introduce you to some of the challenges that are still being a hot topic in this area of research. And then I will pass it on to the mentors in the program that will speak about some of the technical aspects, including some of the case studies that we will use throughout the program. So Bioinformatics for Infectious Diseases is a training program designed to introduce students, clinicians, and researchers to the role and the applications of bioinformatics to the study of pathogenic microorganisms and their interaction with the host, which lead to and enable human disease. And our understanding of the pathogenesis of these diseases can lead to meaningful interventions like antiviral drug design or maybe vaccine design. We'll talk about this in just a few more minutes. So in this webinar, what we'll do is we'll use some of the examples that are case studies that we source from high impact publications and extract from their examples of meaningful application of bioinformatics methods of analysis to different types of omics data, including genomic data, transcriptomic data, metagenomic data, and others. And so what I'll do in my part of the presentation is I will give you a glimpse of how some of these different methods of analysis are used and have been developed to be applied to understand and intervene in these kinds of diseases, including some of the additional challenges that are still being researched and how we can leverage the publicly available data sets that are out there to be able to ask these questions of relevance and importance to what's happening right around us. So the infection itself, what is it? And why are we speaking about infectious diseases as a whole category of applications of bioinformatics? So infection is the invasion of an organism's body tissues by disease-causing agents. Their multiplication and the reaction of host tissues to the infectious agents and the toxins that they produce. So an infectious disease is also known as a transmissible disease or communicable disease, and it is an illness that results from the entry or the actual infection and replication of that pathogen. So the infections that are caused by infectious agents called pathogens include viruses, as well as agents such as viroids, which are not exactly viruses, bacteria, fungi, parasites. And then they are different from non-infectious diseases, which are also recognized as chronic and can be caused by such factors as the environment, nutrition, lifestyle, as well as genetic inheritance. So we typically think of them as two separate categories, but as we will see today, they actually have a lot in common, and one can be the result of the other. So the replication of pathogens in the host leads to major interference, starting from membrane disruption, as in the case with viruses, and the release of toxins, as the case of many bacterial pathogens. Many of these interferences include molecular biological pathways that are designed to fend off intruders and signal to neighboring cells of danger. The pathogens can modulate these pathways and cause long-term organ damage, such as lung tissue scarring due to the immune hyperactivity. So understanding of these biological mechanisms, the way that the entry happens, the way that the body responds, the way that the viral and bacterial pathogens can take and modulate those are all the subject of bioinformatics for infectious diseases because most of this information comes from data. So let's take a look at some example publications to see how this has been monitored and studied using different types of omics data. So the first example I want to bring up is the outbreak of Ebola virus that happened in the 2014-2016 outbreak in West Africa and led to almost real-time genomic surveillance of this disease. As a result, genomic technologies were shown to be effective and useful in an outbreak of an epidemic and led to significant interventions like the discovery of antiviral compounds and design of vaccines. And so as a result of genomic surveillance, better understanding of the disease and intervention was a meaningful success that shows how valuable genomic data is, and therefore bioinformatics, which allows us to study this kind of genomic data and find meaningful insights in this data. Another example that I have here is an example from SARS-CoV-2. So obviously living in the pandemic, the role of genomic surveillance allows us to answer key questions about where did the virus emerge from, how is it developing over time, and how can we anticipate future directions for this viral replication and evolution in order to be more effective at the types of interventions that we can implement, including the testing of whether standard interventions that we think are effective or advanced interventions that we might not think are effective at all could be applied in a meaningful way to contain this pandemic. Another example that you can see here is the genomic surveillance of Plasmodium falciparum, which is malaria that is endemic in some areas of Africa. And still to this day, after millions of deaths, has not been addressed using a vaccine. So to understand this specific pathogen, genomic surveillance is being used, also to understand various factors that lead to greater transmission, replication, severity of disease, and as a result, we can see how interventions are being developed and tested that are becoming more and more effective to contain this disease. Another interesting article that is fairly recent is the increased serological response against human herpes virus 6A, which is associated with risk for multiple sclerosis. This idea has been proposed back in the 1990s, but only recently have there been validated studies that show that indeed there is a statistically significant association between human herpes virus and the development for multiple sclerosis, which leads us to think about many other types of diseases that we think are purely neurological or perhaps genetic and a result of somatic mutations like cancer, but in fact are caused by different types of infectious agents, pathogens. For example, here are the 7 known viruses that cause human cancers. So an estimated 12% to 20% of cancers worldwide are thought to be resulting from tumor viruses. So we're not only speaking about infectious diseases that can be monitored and studied using omics data, we're also speaking about other diseases that we haven't asked about what caused through, but those associations are started to be important to understand. So those were viruses. What about bacteria? Bacteria are another pathogen. Some bacteria could be pathogenic. And interestingly, you can see this article here that was from 31st of January in 2022, the staggering death toll of drug-resistant bacteria, which talks about a global survey that shows that in 2019, antimicrobial resistance killed more people than HIV or AIDS or malaria. So not only do we see that viruses, parasites, but also bacteria is a major cause for global health. Not only that, we also see that drug-resistant tuberculosis poses a major risk to global health security. And we all hear about antimicrobial resistance in hospitals of new superbugs that are not even known how to be treated. So this area of research is not only interesting, is not only relevant because of the pandemic that we're living through, but it is also a major direction for anyone who's interested to apply bioinformatics skills and data analysis skills to address major challenges that we face today and will continuously face in the future. One of the interesting things about the different pathogens that we discussed is that naturally our body already has a response to many of these infectious pathogens. Hosts can fight infection using their immune system. The mammalian hosts react to infections with an innate response, often involving inflammation, followed by an adaptive response. Vaccines are used to train the immune system to identify pathogens and store the necessary epitopes for antibody production in the long-term immune memory. But due to a variety of factors, such immune responses can be insufficient on their own. Two main approaches that have significantly reduced death from infectious diseases in the past century are the antibiotics, antiviral drugs, and vaccines. In fact, if we look at the timeline, we can see that the vaccines have been discovered first, but many other ways to intervene have reduced significantly the risk that infectious pathogens cause or pose. So here you can see from the 1900s all the way to 2000s, this is the rate of infection, which is per 100,000 population per year. You can see here simple interventions. For example, health departments, establishment of health departments, or continuous municipal use of chlorine in water in the United States. You can see the kind of how specific diseases that we think maybe are, well into the past have actually not been so long ago. So this is the influenza, the flu pandemic that killed millions of people, plague that was last observed here in the 1920s. The use of penicillin, if you think of penicillin as something that was discovered long ago, you can see that actually that was in the 1940s, so not that long ago, and the first vaccine that was introduced in the 1950s. So all of this is recent history. This is the history that many of us have seen our parents go through or grandparents. We know that these things have happened very recently, and a lot of the developments that we are using today are based on these kinds of interventions. But since we see such tremendous decrease in these kinds of pathogenic diseases, one might think that, well, we've mastered the way to train our immune systems and protect ourselves from these pathogens. But in fact, we see that COVID-19 has proved all of us wrong. Here you can see this is a comparison to different other causes of death, and COVID-19 surpassed all of them, which shows clearly that we have not mastered the art of understanding and being able to effectively intervene with these different pathogens. However, what does give us some promise? So bioinformatics, I think, is one of those disciplines that allows us to stay somewhat optimistic about the future of infectious disease research. And the reason I say that is because with COVID-19, what we have seen is the continuous application of bioinformatics and data science to practical interventions that are being used in real time and are being used to adapt a response quickly to this dramatic threat that we now understand much better, and we know how to intervene much better as well. What is bioinformatics? Bioinformatics is the intersection of computer science, biology, and statistics. And what I mean by that is that we are able to apply algorithms and software, rely on databases of different types of data, and use these data science skills to be able to analyze data. But this data isn't meaningful unless we understand the biological context. And so the biological context, the specifics of infectious diseases, the pathways, the different pathogens, the biology of the interaction between the host and the pathogen, all of those are a critical component for us to be able to ask meaningful questions and find answers that are significant and relevant to the type of data. Now, in the middle of that are statistical and machine learning or analytical skills that we need. And those analytical skills primarily are driven by our ability to structure data and then ask questions that are generalizable. These questions are not only relevant to a particular study, they're relevant in general to some biological phenomena. So one of the important things in the context of infectious diseases is that particularly in this area, there has been a dramatic increase in the number of data databases and data types that are available for infectious disease research. So here you can see, this is a slide from maybe a few months ago, but you can see here that the rate of discovery of viral genomes, the way that these databases have become structured and accessible is increasing dramatically. And that means that we can start asking meaningful questions about all of these types of questions that we might have that we just learned about in a fairly easy and user-friendly way. And so the purpose of training in the context of bioinformatics for infectious diseases is to start thinking, how can I ask an important question? What kind of examples can I follow? And where can I find data sets that provide me with sufficient answers that I'm looking for? Because even though so much has been already done, not everything is clear. In fact, we know that the diseases that are caused by pathogens vary significantly between individuals, locations, or even whole populations. They happen at different speeds, at different severity, and the understanding of how they happen is linked to a very deep understanding of the immune responses as well as the evolution of these pathogens. And not only that, we also are now discovering that many of these viruses and emerging pathogens are actually not only in individuals, but they also are in the environment. They're all around us. They're in other animals and other species. And so we have to expand our understanding of this molecular biology to also be able to detect specific types of pathogens like viruses in other environmental samples that we might not have thought of yet. And that leads us to this major challenge of annotation and curation of data to understand relationships between strains, species, and phyla of different pathogens, and our ability to then understand their function. How do they function? How do they interact with the host? So in the context of this program, one of the key technologies that we're going to be using and we're going to be talking about a lot is next generation sequencing. Next generation sequencing could be used in many different ways. It could be used to analyze individual genomes like the COVID-19 genome. It could be used to analyze metagenomes where we have a collection of genomes in a particular sample and we need to understand diversity or maybe relationships between these genomes. And also we need to understand the different types of library preparation techniques and steps to prepare the data to make sure that we can answer specific questions. Now, to be able to use this data, we need to understand the biology, we need to find the right kinds of data, and then we need to know a variety of techniques that will make it easy for us to interpret and find meaningful associations. And that includes understanding of viral genomics and evolution, or phylogenetic. How can we build phylogenetic trees and monitor progression over time, connecting it with temporal or maybe different types of phenotypes that we have? Understanding of transcriptomic data, or how to find differences in gene expression and compare and contrast different types of conditions or groups of samples. Variant calling, where we can find variants in specific positions on the genome and be able to interpret them. And analysis of metagenomic data, data that contains thousands or even millions of different microorganisms that need to be categorized and found relationships between them. The examples I'm sure that you have seen in the news and elsewhere, but the examples that we will use in this program include such epidemics like the Ebola virus, which you can see right here, the understanding of origins, transmission, replication, and development of continuous evolution in this pandemic and how it affects our body's ability to respond to infection and produce relevant immune responses, differentiating between specific processes and stochastic processes, and then linking this understanding with our ability to take a sample and monitor the specific types of clades and variants that are of concern. And ultimately being able to translate this information to report on our findings, communicating the results effectively with meaningful annotation and interpretation. So to summarize, what we will do in this program is we will take a look at different types of omics data, including genomic, transcriptomic, and metagenomic data. We will learn to deal with these data sets, align them, find associations using different types of processes, interpret them using our understanding of structure and function of different genes and proteins, and then measure host responses to really understand how does the host respond to the different infections that are there. Now, data from next generation sequencing is very complex. This is an alignment of one of the first sequencing data sets from the SARS-CoV-2 samples that were identified in China. And so as you can see here, it's not trivial to get from this kind of complex information to some meaningful insights. And so what we will try to do is we will try to break it down to understand the principles behind the data that we have, the analysis methods that help us to perform some of the functionality in a semi-automated way, link that with biological understanding of the processes that drive the observed variation that we have in the data, and then use analysis methods that help us link those into meaningful models behind the process that we think is driving this variation. Now, ultimately, we will also talk about ways to interpret such data, not only find specific variation that is meaningful, but then also use the appropriate route for interpretation. And we will use different examples to link this kind of sequence variation to actual interaction and function of different proteins that anyone can do independently once you find the relevant, meaningful differences in your datasets. Now, to speak about specific examples of how this type of bioinformatics approach can be used to study viruses, bacteria, and different types of pathogens, I want to invite our mentors that will be helping you work through these different case studies, apply these different technical methods, and find meaningful insights from relevant biomedical publications. I want to introduce you to our mentors. Dr. Raghavendran is going to be your research consultant and mentor. He's going to be helping you understand some of these topics and guide you in the specific case studies. Dr. Priyo Pal is going to be a teaching assistant that will help you understand any kind of technical issues that you will have throughout this training. And Dr. Mohit Mazumdar, who's also a project mentor and an expert in computational biology, who will talk about how sequence and structure poses some interesting insights and future directions for this kind of research. So Dr. Raghav, I want to invite you next to talk about some of the examples that we discussed.
Thank you. Thank you very much, Elia. Let me find a way to share my screen and yeah. Yeah, I hope my screen is visible to everybody.
Yeah,
Yes, Dr.
Okay,
Raghavendran.
perfect. Yeah, I also got confirmation. Hello, everybody, one more time, and I'm Raghavendran. I'm a research consultant and mentor with Pine Biotech and Omics Logic. I'm trained in bioinformatics and I hold a PhD in computational biology from Max Planck Institute, Germany. My broad research interests include are in the application of data science methods to understand cancer biology. My role in Pine Biotech includes developing curricula for the platform and also mentoring students in the research fellowship program and also in the programs that you'll go to sign up. Please allow me to talk about few example projects that we have prepared in the platform Learn Omics Logic platform that will help you appreciate how bioinformatics analysis can be leveraged to understand the biology of pathogens and to study infectious diseases. So first, we will focus on a project that studies the current pandemic, COVID-19. So in this project, we explored the pathogen SARS coronavirus itself and as well as few diseases that are caused by it. So this project will help you explore the origin of the pandemic and how different diseases are caused by this pathogen and how they are related to existing strains from human coronavirus, and also to other strains from animals sources also. So we will do this by using publicly available data from repositories like NCBI virus and GEO data, etc. So we will learn in this project, we will learn to perform or how to perform multiple sequence alignment followed by phylogenetic analysis. These analyses will help us compare the existing strains of coronavirus to understand the zoonotic origins of the disease. So we will also learn in detail how to interpret results from analysis like multiple sequence alignment to understand mutational changes in the genome of SARS-CoV-2 compared to different strains, as I have already told, strains from humans and also strains from different animals. Next, we will also see what are the important protein components of the virus through which they enter into the human cell. And we will also learn how to visualize the identified mutations on the spike protein, for example. And from such analysis and such visualization, we can understand and we can study which mutation might affect the functional properties of this spike protein and alter its ability to bind to the receptor in the host cell. So furthermore, we will also learn how we can look for transcriptomics data from patients that are infected with SARS-CoV-2 using public repositories like Gene Expression Omnibus database and etc. Here, we will explore such data, this data in particular, to find out important transcriptional signatures that are associated with drug treatments like ruxolitinib, RUX in short. We will also learn some important bioinformatics methods that are available for identifying gene expression signature for treatments like RUX or ruxolitinib. We will perform differential gene expression analysis between RUX treated and untreated samples. And we will also employ unsupervised machine learning methods like principal component analysis and clustering to understand the potential of these signatures in categorizing drug treated and untreated samples. So, during the course of this project, you will learn how to do and the logic behind such analysis and how to interpret these analysis in the context of the data or in the context of the biological project or question in hand. So next project we will get introduced to is on the outbreak of enterovirus D68 or EVD68 in short. This is a virus that causes a severe neurogenic disease called acute flaccid myelitis, AFM in short, mainly in children. So it is in comparison, it is a polio-like illness defined by the acute onset on spine or on the paralysis of the spine. So in this project, we will understand the evolutionary relationship between the strains of different outbreaks of this virus. So to go into the history, this virus first emerged around 1960s. And in the last decade itself, the outbreak of EVD68 has been reported across different parts of the globe, even in developed countries like United States, Europe, and roughly once in 2 years or once in 3 years or so on. So their pathogenicity also varies among these outbreaks and among different populations of this virus. So similar to the SARS project, here also we will use various phylogenetic tree reconstruction techniques and algorithms to find out whether the EVD68 strains from different outbreaks at different time points like 2014, 16, 18 are from the same virus or from the same strain or from the different strain or a related virus using next-gen sequencing data of the patients. So we will also see that phylogenetic analysis reflects the variations in the strain that causes these outbreaks in different years or in different time points. Furthermore, multiple sequence alignment will reveal a number of variations in the sequences of different viral strains. A careful analysis and how to perform this careful analysis will help us identify critical mutations that can cause significant changes in the amino acid level among these different strains. These mutations can be related to the virulent properties of the pathogen and will help us characterize the viruses like EVD68 or SARS-CoV-2, which is currently a very dangerous pandemic going on, based on their pathogenicity. Hope that was informative and that was very interesting. And I look forward to meet many of you in the Bioinformatics for Infectious Diseases program. I thank you very much for your attention and will now pass the stage to Dr. Priya. Thank you very much.
Yeah, hello everyone. Just give me 2 minutes to share my screen. Yeah, so can you see my screen?
Yes, ma'am, we can see your screen.
Okay, so I'm Dr. Priya Uppal. I'm a research and curriculum coordinator at Pine Biotech. I did my postdoctorate from Indian Institute of Science, Bangalore in molecular biology and bioinformatics. My PhD is in life sciences from GNDU Amritsar with specialization in stress physiology. And I have 25 publications so far, which are published in journals of national and international repute. Regarding my, just give me 1 second. Sorry for the interruption. Actually, there was some network connectivity issue. Regarding my research interest, it is in next generation sequencing, cancer biology, drug discovery and repurposing, stress physiology, and data science. So I'll be talking about the Ebola virus outbreak. So we all know about the 2014-2016 outbreak of Ebola virus, which occurred in West Africa and it affected thousands of people all across the world. We all know that it is a viral disease of humans and primates, and it is caused by Ebola virus. So to develop the vaccines and antiviral drugs for this infectious disease, we need to understand what is the origin of this virus, what is the shape and structure of this Ebola virus. So I've designed a project on this Ebola virus, which is based on original publication data to understand the origin and to find out what are the important mutations or the variants which cause this lethal outbreak. So we got the data from NCBI platform, NCBI virus platform. Here we used genome sequence from this particular platform, and we performed multiple sequence alignment and phylogenetic analysis to determine the evolutionary relationships between these genomes. And then what we identified was we identified the groups of samples and saw how they are related to the phenotype. So we used TBio platform server, which Sonalika and Ilya and Dr. Raghav, they've already spoken about this. So what we analyzed mutations between groups of samples using comparative analysis to study the mutability. This method helped us to identify the origin of polymorphism, which is believed to cause an increase in the ability of the virus to enter its host cell. So this is all from my side, and I'm looking forward to the participation of all the participants in maybe upcoming the research fellowship program. So thank you so much. Over to you, Sonalik.
Thank you so much, Dr. Raghavendran and Dr. Priya. Now, I would like to invite Dr. Mohit to help us understand the structure, the program structure of bioinformatics for infectious diseases. Over to you, Dr. Mohit.
Thank you, Sonalika. Am I audible?
Yes, sir.
Okay. So can you see my screen also? Sorry for asking.
Yes, sir. No, it's perfectly fine, sir. Yes, we can see your screen.
Hello, everyone. It's great to meet you all today. I'm Mohit, and I have a PhD in computational biology and specialization in molecular modeling, simulations, and discovery related research projects. So in today's session, in a few slides, I'll go through the publicly available data, recent structural data from the pandemic. So talking about that, the structural biology response to the COVID-19 pandemic has allowed the world to see the virus, the virus is attacking us in a structural perspective. So we can visualize, we can see the structure. So it has also aided in vaccine and therapeutic development and has shown foresights in the investments in structural biology capabilities in techniques such as X-ray crystallography or techniques such as cryo-EM or the MX. So this sheer magnitude and uniqueness of the event that has that has enabled all of us in a way that helped in all these different types of data that has come out from different repositories. So today we are going to get an overview of where this data is and how it looks. So the speed with which these structural investigations have occurred has been really breathtaking. Simultaneously, the availability of these data has benefited from the long history of making these repositories and the metadata that is also available with this information about the structural information, and all of this is available in repositories such as Protein Data Bank, a worldwide repository for macromolecular structures. And also we are going to talk about some other aspects of getting structure, which is through molecular modeling. So as there is like this 1.5 year since the declaration of COVID-19, and recently 1,000 structures of SARS-CoV-2 biomolecules have been deposited in the PDB itself. So these are all experimental structures that have been deposited in Protein Data Bank. So it is like kind of rare to obtain a snapshot of such immense effort in structural biology, and this happened due to this revolution of not only the need or the structures, but also the technology. So with cryo-EM, there has been a lot of insights that we got from the resolution revolution. That is what it is said in aspect of cryo-EM microscopy. Talking about the computational aspect of this. So first, the experimental aspect is about the structural biology events and experiments that led to the information of this kind of data, that how these structures are. But still, there are many limitations when we are talking about the complete understanding of how it relates, how structure can be related to the function. So there are, that's where, I mean, computational structural biology or computational structural bioinformatics comes in. So where we are talking about, modeling complex structures to gain insight, or to be able to understand the effect of mutations on these experimental structures. So those things can be modeled and have been playing a very important role in understanding the pandemic more and more, and to find solutions. So the SARS-CoV-2 pandemic, as I said, has contributed a lot in terms of also structural biology work. So in silico structural biology work. So initially what people were used to do is, I will tell you about some of the softwares that are available, online tools, such as the one that is listed here, AlphaFold. But also there is a software that many of us use for structural predictions is CI TASR. So from Zen's lab. So it's an online available tool. And also AlphaFold from DeepMind Consortium. They have been able to, with these tools, we have been able to leverage the release genomes, determine protein sequence, and also to create computational models. And this has been happening till the early March 2020 to till date. So some modeling, a lot of insights that we could draw from structural data is via modeling. So how do we model complex structures? How do you model single point mutations or multiple mutations and understand the variants? And also then there is an aspect of molecular dynamic simulations that have massively also extended the utility of experimental structural data, that helps in providing insights via the rapid analysis of these in silico mutagenesis for many key viral proteins, and also the ability to, for example, model glycosylation of the spike protein. Our computational approach has also helped in probing host pathogen protein interactions, so protein interactions, and then contributing also in network based and machine learning based analysis. And we have an upcoming talk about the network analysis tomorrow that I would like you to all know about Sciomex Club. So we will be sharing that information also with you. So about the structures, and to understand like what is available out there and how to understand and make use of this. So that is an important aspect of integrating structural biology into your research, or utilizing the data that is available publicly. So the spike protein of this virus, I mean SARS-CoV-2, is one of the central figure, right, in this fight, since it is the primary target for antibodies to provide and to target of antibodies that provide immunity against the virus. So the surface of coronavirus coronaviruses, I'll give you a layman explanation. So the surface of coronaviruses are covered with these spikes, giving them their distinctive clown-like appearances in the electron micrographs. So many people are already aware of this, and I think most of us are. So the spikes initiate the process of infection, binding to the receptor, and then fusing with the cell membrane to release the viral genome inside. That's the process. So many other, and it has been seen in many other models as well, like influenza, hematoglutinin, and also HIV and Ebola. So the spike protein is composed, of three identical chains that form together a complex with a small domain inside, which is inside the virus, which is the membrane spinning segment, and then there's a large ectodomain, and that all you can see some of those structures that you can see in this slide. So these structures also include a portion of the many sugar chain that coats the spike. Since the sugars are highly flexible and difficult to observe, that's one of the challenges. So many things that you can gain insight from are from the structures. And to tell you more about that, so recent structures have revealed that the spike of SARS-CoV-2 and SARS-CoV and SARS-CoV-2. So comparing both, when we compare both, we found out that they are quite flexible. So in these structures, the receptor binding domains, the receptor binding domain are seen in different conformation. So when you compare structures and when you're superimposing them on one another in a three-dimensional space, you can see the differences and how there is a movement. You can find out about that movement just from your screen. So by learning about these different tools that we will be covering in this entire program. So often just one domain, in this case of SARS-CoV-2. So many times what is happening is that one domain, you would see that there is some movement, and then there are several other extensions that you will see that they are tucked down, or there are different type of movements that have been found out in these different types of conformation. And then researchers have hypothesized that the recent SARS-CoV-2 variants are virulent because their receptor binding domains are particularly flexible. So that's one of the hypotheses. And whereas most spread coronaviruses that cause the common cold are less dangerous because they are less flexible. So that's one of the hypotheses that you can understand by comparing some of these structures and understand from the different conformations. That include the binding. To understand this has to be modeled. So you have structures from different conformations that you obtain from different protein data banks, structures which are deposited online, and then you try to model them into a complex. That will give you a clear insight of how this is done. So these are some of the things that you need to learn computationally in computational biology or in silico computational structural biology. So about the receptor binding, our immune system, fight backs, right, when coronavirus infects us. So that's where the antibodies comes in. So antibodies are what? Are protein molecules that play a key role in any organism's defense against a foreign molecule called antigen, right? The first step in this process is specific complex formation between an antibody molecule and a molecule of the antigen. So the structural basis of this antibody antigen recognition is started to getting elucidated from these structures. So these structures are helping us to understand that mechanism, how it works. So the spike is a major target of this protection, right? Since it is exposed to the surface of the virus, and these three protein structures that you can see on the left, which is 3BGF and 2GHW and 6W41. And you can totally search them by their 4-digit keyword. You just paste them on your browser, it will show you to the link to their actual structure that has been deposited in the PDB. So these are PDB IDs. So these three structures show that the antibody can be recognized by spike protein in many ways. So two of these antibody block the receptor binding portion of the domain, but the other antibody targets a cryptic site at the base of the domain that is only exposed when the antibody binds to it. So you see that there are not only just different conformation, but different active sites also that you can find, that you can see, which depends on the movement of the protein. So one thing that you have to understand is that this is a PDB is just a snapshot. And when a protein is in motion, then it is moving. So there are different conformations that a protein attends. And then based on those different conformation, there could be different strategies to design molecule. So this is something that we should consider. These sort of structural information is something that we should consider when we go about, designing a vaccine, or when we are looking for strategies to find out solutions to, finding a drug. So about this, doing all of this is kind of possible. So in this program, we will be learning to use powerful visualization tools such as Chimera, and explore through the available structural data. And then also we will be reviewing several tools developed for your training in this program, to navigate large data sets and then connect the various aspects of, sequence, structure, and metadata into a single experience. This is CNAP. So what it will help us is that it will help us in understanding or addressing many questions. So genomic data analysis can be utilized to see the changes between viral strains. So you're looking at the mutations and different significance level of these detected changes, and how you track them or map them to the 3D position of on their protein structure, so that you can see whether they are on the surface, whether they are in the active site, what could be the role. Then we will be able to do, from sequence to structure. Another aspect is to do comparison, and that comparison at a sequence level could be done through multiple sequence alignment. So multiple sequence alignment is a very powerful tool to understand viral adaptation between animals and protein, and again then trace it back to the structure. Similarly, another aspect of answering some of the research questions would be to understand the relationship between viral and host proteins, such as the trimeric SARS coronavirus surface spike glycoprotein. So this spike glycoprotein binds to a cellular receptor called angiotensin converting enzyme 2, so ACE2. I mean, you might all have heard about ACE2 in short. So, and it mediates through the fusion of viral and cellular membranes through a pre to post fusion conformation transitions. So as I was telling you before also, so because of certain binding, there are conformational changes that enables another binding event. So all of this fascinating stuff, and just imagine to be able to visualize it and to understand from sequence level to a structural level is something that we are planning for this program. And we are hoping that many of you can go through that experience and understand different aspects of how structure and complexes that mapping mutations from a single protein from a population to the three-dimensional structure and understanding the function obviously is something that we're trying to help everyone to learn. So this is what we have planned for the structural part, and that's what I wanted to tell you all about in today's session. And we will be covering these in much more detail when we go through these different case studies that will be covered in the full training program starting from 14th February. And that's about my part of the presentation. And to tell you more about the entire schedule of this program and how the sessions will be and how you could all register, I'll now hand it over to Ms. Sonalika.
Thank you so much, Dr. Mohit, for such a wonderful presentation. Now, I would like to take you all through the registration page to get yourself enrolled for the program commencing on 14th of February. And this is the link to the program page. I'll be pasting it up in the chat box. Also, we'll be mailing out the link to the program page along with the link to the recording of the session to you all after the session. So once you are here on the program page, you'll find all the details that are related to the program. That includes a program overview and most important of all, the schedule for the program. So here you'll find all the session dates and also the sessions. Let me just zoom in, sorry. Yes, here you will be able to find all the session dates along with each and every topic of the session, and also the topics that are supposed to be covered in the sessions along with the associated online resources. So for each and every session, there are some online associated resources that includes the links to the lessons on the Omics Logic Learn portal, and also some mentor guided resources that you will be supposed to go through along with each session. Once the session is completed, once each session is completed, we'll be sending you out the link to these associated resources and also the recorded sessions. In case you miss out on the session or want to revise the session, you'll get the recording as well as the associated resources and publications that are to be followed along with the session itself. Now, when you scroll down on the program page itself, you will find 3 levels to which you can refer to enroll yourself for the program. The first level is a beginner level, which includes the access to live Q&A sessions, group sessions, and also the recordings, the access to Omics Logic Learn portal, and the Bioinfo server educational pipelines. Then we have an intermediate level, which will give you an access to one-on-one mentor support. And then we have an advanced level where you will get some extra time to figure out a research proposal with the help of the mentor. You'll be able to explore some of the data sets from the big data repositories, and you'll be able to come up with a research proposal that you can further work upon in the research fellowship program and get yourself a publication, or at least present your research work in a poster presentation format. That is in the upcoming symposium that is scheduled for March 31st and April 1st. So these are the 3 levels, beginner, intermediate, and advanced, that you can opt for. And you simply need to click on the sign in and checkout button. And you will also be given the access to the early bird discount coupon code via emails, I believe. So our marketing executive Sparshthar will send you a coupon code that will give you the access to the early bird offer, with which you can definitely enroll in the program, picking up any of the levels that you would like to enroll for. And when you scroll down, you will also get the list of program, course, and project library that includes the associated coursework that you are supposed to go through as a part of the program, which includes Omics Logic courses and example projects, which will help you understand how the key concepts, methodologies, and the pipelines that you went through could be applied to answer a real-life research problem and get some hands-on practice. And yes, definitely, many of you had a question about the certification process. So once you successfully complete the program, you will be accredited with a certificate. And this was all the information that was required for you to know about the registration process. And we have the link shared with all of you. Now, if any one of you has any questions, please put them up in the chat box so that we can answer. Okay, so Mohammed is asking, is there any program on analyzing cancer data? I would like Dr. Mohit to take this question specifically, because I think he's the best person to answer it nicely. Over to you, Dr. Mohit. Okay, so Dr. Mohit, I think, is not here. Yes. So when you say cancer data, specifically you are referring to the oncology field, right? So yes, we definitely have a lot of programs that includes the analysis and the understandings first of all, and then definitely to get some hands-on practice with this type of data. So when you say cancer data, or let's say the oncology field, then comes the role of either transcriptomic data analysis or genomic data analysis that you can go through and learn about, right? Or even metagenomic data analysis could be explored and learned about, as we have had recently a lot of publications telling how metagenomics data could be explored to understand the patterns that are responsible for causing a cancerous condition. So yes, so when you say oncology, then we have 3 programs specifically, that is genomic data analysis, metagenomic data analysis, and also the transcriptomic data analysis. And having the knowledge of these particular skill sets, you can definitely apply these skill sets to the specialization track of oncology. And Mohammad Uzair Khan, I'll recommend you if you can just simply put your mail ID in the chat box, and you'll get the list of these programs that you can explore. You can see what all topics these programs includes and what exactly would you like to explore and learn from and get some hands-on practice. So just put your mail ID and we'll be able to help you out with that. Omuvami is asking how many hours are the session and the time, please. So Omuvami, the sessions will be an hour and a half each, though they are scheduled for an hour, but because we have discussions with the program participants, definitely it gets extended to an hour and a half or maybe 2 at max. And these sessions are scheduled for 9 a.m. CST. You can convert it to your time zone and see what exact time it becomes. And also, let's say you are not available at 9 a.m. CST and you're not able to make up to the live session, then you can definitely go through the recorded session and take up your questions in the Slack channel. So we have Slack channel for each and every program that we have, and you can take your questions in the Slack channel. You'll also have the mentors up there and follow the recordings specifically in that way. And for the session schedule, please refer the program page here. You have all the dates listed, and also you have all the topics listed for each and every date. Dr. Raghavendran, you are here with us, right? We have a specific question that I would like you to address. The question is by Saurav Roy. He's asking, I would like to ask if there is any blockchain-based technology available for bioinformatics or any in development.
Ooh, that's a toughie. I mean,